Okay, all right, welcome to the lymphatic system. Okay, the lymphatic system is going to consist of one chapter, and you will have a test for only one chapter. Can you believe that? It's a little out of the ordinary, but it's a little complicated, but only one chapter at a time. This shouldn't be too bad. Okay, let's go ahead and get started. All right, so the lymphatic system consists of basically two different parts, two semi-independent parts. There's a network of these lymphatic vessels, and then there's lymphoid tissues and organs that are scattered throughout the body, including lymphatic nodules, lymph nodes, tonsils, your spleen, and your thymus-spleen. Okay. It also returns interstitial fluid and leak plasma proteins back to the blood. Lymph is the interstitial fluid once it has entered the lymphatic vessels. Okay. Functions of the lymphatic system. Okay. Maintaining fluid balance in the tissues. It also helps to return 3 liters of fluid a day back to the circulation, back into the blood. Okay. It also absorbs fats from the small intestine and in the lacteals, which are specialized lymph capillary cells, or capillary veins, okay, that are present in the intestinal mucosa. And these lacteals absorb digested fat and deliver chelae, okay, fatty lymph back to the blood. Okay, it also helps defend against microorganisms and foreign substances. The lymphatic system is a one-way system. It's pumping through and then it's pumping back to a certain place. It's just going from one direction through the body. Lymph always flows toward the heart. Okay, it's always returning towards the heart to return to the circulation. Lymph vessels include microscopic, permeable, line-ended capillaries. Okay, line-ended capillaries. Where they terminate. Okay, line-ended capillaries. Lymphatic vessels and ducts. The lymphatic capillaries are in lymphatic vessels. Okay, the lymphatic capillaries give lymphatic vessels and then lymphatic ducts. Okay, they increase in size as you work through this progression. Capillaries are smallest, vessels are a little larger, and ducts are much larger in size as you move along. So here are some of the major organs of the lymphatic system that we'll look at. Let's look at a few of these real quick. Okay, to start off with the thymus gland. Okay. Thymus gland is found right in the center of the chest. Okay, it should be fairly familiar because we've looked at the thymus gland a little bit before when we were talking about endocrine glands. Okay, axillary lymph nodes. Okay, you're found right in here. Found different areas around the arms and different places like this. And in females, especially, you have a congregation here called the mammary plexus that is located around the mammary glands as well. Intestinal lymph nodes, okay, lymphatic ducts, okay, you got different lymphatic vessels, and then major area right here in long bones you have bone marrow, which is also part of the lymphatic system. It's part of the circulatory system. It makes blood vessels. Okay, it's all connective tissue, but it's also lymphatic because it produces the white blood cells as well. Okay, here in the throat we have the tonsils. Okay, and the tonsils are major lymphatic organs that help defend your body against foreign invaders. Okay, you also have cervical lymph nodes in your neck. Okay, which you feel often when you get sick. They swell and fill with basically white blood cells that are in the lymphatic system. Then you have thoracic ducts, okay, which are connected to subclavian veins. And then moving down through the body, you have your thoracic duct right in the center line and midline of the body. And then you have one of your major lymphatic organs, or should I say your major lymphatic organ here, your spleen. Okay, the lacteals of the intestinal wall, and then your inguinal lymph nodes. What is inguinal lymph nodes? Alright, next question. Number 1. What are inguinal lymph nodes? forget this part. Sorry about that. All right. Lymphatic capillaries and lymphatic vessels. Make sure I skip the slide. Sorry. All right. Lymphatic capillaries are similar to blood vessels, but they have modifications. Okay. They are very permeable. You know, blood vessels are not very permeable, except for the capillaries, and very little can get through the capillaries because they have such small openings. Well, lymphatic capillaries are much more permeable. They're loosely joined endothelial mini valves, basically, so they can open more widely, larger cell valves. And they also withstand interstitial pressure and help them to stay open. Lymphatic vessels also have valves that ensure the one-way flow of lymph. Okay, lymph, which includes a lot of the disease-fighting parts that you need in order to fight off infection. Lymph is moved through contraction of lymphatic vessels, smooth muscles, skeletal muscle action, as well as thoracic pressure changes. Lymph formation and movement. Okay, so here as the blood leaves the arterioles and moves into the capillaries, okay, you begin to have the formation of lymph. Okay, because it starts to leach out from here in these regions here in the capillaries, and the regions from the capillaries then leach these cells, and these cells enter into the lymphatic vessel system, which is this right here, lymphatic vessel system. Okay. Lymph is delivered into one of two large trunks. Either the right lymphatic duct, which drains the right upper arm and the right side of the head and thorax, or the thoracic duct. And thoracic duct simply arises from the cisternal chile and drains the rest of the body. So you've got the one that's draining the head, only the right side of the head, and your thorax. Okay, basically your chest region, your right lymphatic duct, where the thoracic duct does most of the rest of the body. Lymphatic tissue. Reticular connective tissue contains lymphocytes and other cells. It also can be surrounded by a capsule, either lymph nodes, spleen, or thymus gland. Okay, and it can be non-encapsulated. There's other areas that are non-encapsulated, like lymphatic tissue, lymphatic nodules, and your tonsils, which are not encapsulated at all. Okay, diffuse lymphatic tissue consists of dispersed lymphocytes. Okay, lymphatic tissue consists of the lymphocytes themselves. Remember talking about lymphocytes at all when we talked about white blood cells. Okay, remember that these are white blood cells that have left the blood circulation and have now moved to the lymphatic system. Okay, and it has no clear boundaries. So it can grow large or smaller. It has no boundaries. Okay. There's no membrane around these lymphatic tissues. Lymphatic nodules are small aggregates of lymphatic tissue. For example, your Peyer's patches. Okay, the Peyer's patches are found in the small intestine, and they consist of large groups of lymphatic tissue. Okay, more about the Peyer's patches. Okay, the Peyer's patches consist of isolated clusters of lymphatic tissue, which are similar to the tonsils that are found in your throat. Okay, but they're found in the wall of the distal portion of the small intestine. You know, the part that's moving. You know, it's not the part up top where it's starting to digest. It's where it's working down, and then it starts working with the colon. It's where it meets the colon. Okay, the similar structures are also found in the appendix. Okay, the appendix, remember that gland that for years people thought, well, you don't need that. You can just remove it and be fine. Well, we found that there are other uses, and now we're finding that it has direct links to the health of your body through its ability to help fight infection, and in fact it actually stores gut bacteria in certain animals and certain populations not so much in the naturalized world where we're exposed to antibiotics. Okay, the Peyer's patches and the appendix. Okay, Peyer's patches and in the appendix, they destroy bacteria and prevent them from breaching into the intestinal wall. So the Peyer's patches play a role in keeping your digestive health and your overall health intact by helping defend against bacteria. And they also generate memory lymphocytes for long-term immunity. Okay, so now we're finally starting to get to a little bit of memory for invaders coming into the body. First, associating it here with memory lymphocytes that are found in the Peyer's patches. Okay, so here we can look at the lymphatic nodule. Okay, and we can see the diffuse lymphatic tissue within. And this is under a light microscope at a magnification of 25x. Okay. As you can see here, we have many areas. Whoa, sorry about that. Was not expected to go back that far. Okay, the areas of the lymphatic tissue. You can see the large gaps and the caniculines, basically, in that area. Okay, tonsils. Let's go ahead and talk about the tonsils. The tonsils are the simplest lymphoid organs, and they form a ring of lymphatic tissue around the pharynx. Okay, right back in here, in the throat. If you look at your tonsils right at the back of your throat, right past your flap at the top. Some people call it goose. What do you actually call that? That's going to be question number 2. It's an odd question number 2, but it's still question number 2. Okay, I don't know how I did this. Sorry about that. Question 2. What is the tissue that hangs down in this type of shape in the back of your throat? What do you call that? What do you call that piece of tissue? Alright. Forms a ring of lymphatic tissue around the pharynx in the back of the throat. Okay, the location of the palatine tonsils is on either side of the posterior end of the oral cavity. What is the oral cavity? Your mouth, right? So they're found on either side of the root of the mouth, posterior end of the mouth, back here. Okay, the lingual tonsils lie at the base of the tongue. The base of your tongue, it's not quite in your mouth. The base of your tongue goes down into your pharynx, into your throat. Okay, the pharyngeal tonsils are found on the posterior wall of the nasopharynx. Okay, the nasopharynx, remember the nasopharynx, where your throat connects to your nasal passage. So they're found just a little bit higher in the back of your throat. Okay, the epithelial tissue or skin tissue that overlies the tonsil masses invaginates. Okay, in other words, it forms openings, forming blind-ended crypts. And these crypts tap and destroy bacteria, in particular types of matter it finds foreign, which is almost anything that you would imbibe in this way. Alright, so now let's look at the pharyngeal tonsils. The pharyngeal tonsils, those that we find in the throat at the back, but in the area near the top of the mouth. Palatine tonsils, interior the sides of your mouth. Okay, and then your lingual tonsils, the ones found in the back of the throat, to the front side of your throat, and to your portion where your tongue comes down into your throat, at the very back or base of your tongue. Okay. Lymph nodes. Lymph nodes are the principal lymphoid organs of the body. They're found in lots of different places. They are very, very spaced out throughout the body. They're embedded in different types of connective tissue and are clustered along lymphatic vessels. So they form a way in which cells from lymphatic vessels or material in lymphatic vessels can coagulate, group in certain areas, or come together and form an area to help fight an infection. That's why the lymph nodes swell during the infection. Okay, they're also embedded in the connective tissue, as I mentioned before. And aggregates or groups of these nodes occur near the body's surfaces in the inguinal, axillary, and cervical regions of the body. There's two basic functions. Okay, the lymph nodes will filter. Okay, macrophages. You're attacking white blood cell types. Will destroy microorganisms and debris, but remember they're not white blood cells when they're in the lymphatic system. They're lymphocytes. Okay, and then they also provide the ability to activate the immune system by monitoring for antigens and mounting an attack against these antigens when they enter into the system. So let's look at a lymph node. Okay, the lymph node is made up of multiple areas, and let's check these out. Okay, the main two areas that it separates into are the cortex and the medulla. Okay, so here we look at the cortex, and we find that we're composed of diffuse lymphatic tissue, lymphatic sinuses, lymphatic nodule, and the germinal center. Okay, and then over here on the right-hand side of the screen, we see the medulla. Okay, and it's composed of diffuse lymphatic tissue, just like the cortex is, but it also has lymphatic sinuses. Okay, remember the sinuses are gaps, so those are areas in which things can be stored, or things can be moved through. Okay, lymphatic nodes. Okay, lymph nodes. The nodes are bean-shaped and surrounded by a fibrous capsule, and the trabeculae extend inward from the capsule and divide the node into different compartments. And these nodes have two histologically distinct regions. There's a cortex and a medulla. Okay, medulla, medulla. You know that's going to get thrown around both ways. All right, those two regions, as I mentioned before, they're going to be really important to learn the difference between later. Okay. Cortex contains follicles with germinal centers. These germinal centers are heavy with dividing B cells. Okay. There's going to be a couple different types of cells that you're going to learn, and B cells are going to be one of the main ones that function to fight infections and help with immunity. There's going to be B cells and T cells, but go ahead and remember the B cells first. Okay. Dendritic cells nearly encapsulate the follicles. The follicles are found almost surrounded by dendritic cells. Okay, and then there's the T cells that circulate among the blood, the lymph nodes, and the lymphatic stream. Okay, T cells or killer T cells are also very active with immunity. And deep cortex will house T cells which are in transit from one area to another. Okay, the spleen. The spleen is found in the left superior side of the abdomen. Okay, and there's two distinct areas. There's the white pulp region of the spleen, which contains mostly lymphocytes suspended on reticular fibers, and then they're involved in the immune functions. Whereas you also have the red pulp, and the red pulp consists of the remaining spleen tissue that is concerned with disposing of worn-out red blood cells and blood-borne pathogens. So the spleen is playing a dual role there. It's cleaning the blood, and it also has the white pulp region, which is containing most of the lymphocytes. The spleen is also a limited reservoir for blood. So like I said, it's got a dual role, both in the maintenance of the blood and as a lymphatic organ and keeping up with the lymphocytes. So here we look at what's going on here with the spleen. or kidney. I got mixed up there. Oh, yeah, I was talking about spleen in the previous video. So here we're talking about renal surface, splenic artery, spleen. It's been a long week. Alright. Branches from the splenic artery are surrounded by diffuse lymphatic tissue. Alright. And the arterioles enter a different nodule that divide. Okay. So you can see how it comes in here through the arteries. Okay, and it moves around in here, and we can see the inside part where we have the splenic artery branching and moving through the white pulp, and outside the area of red pulp where we have the splenic veins. A few capillaries will directly connect to the venous sinuses. Okay, remember the venous sinuses, where are those found? You see the venous sinuses? Okay, those open areas right there where storage can happen. Alright. All that's going on. And we've got that. Okay, and the ends of the capillaries are then separated from the beginning of the venous sinuses by a small gap. Okay, and then the blood can rapidly cross this gap. And some capillaries empty into the splenic cords. Okay, and then the venous sinuses connect to a branch of the splenic vein. Alright, let's go ahead and move on to the thymus gland. The thymus is a bilobed organ. Okay, it's made up of 2 lobes. Okay, that causes T lymphocytes to become immunocompetent. The size of the thymus varies with age. In infants, it's found in the inferior neck, the lower portion of the neck, and extends into the mediastinum where it partially overlies the heart. So it is very large in comparison. Okay, it increases in size and it's most active during the childhood years. Okay, but it stops growing at a certain point during adolescence and then gradually starts to atrophy or die off as you mature later in life. Okay, it differs from other lymphoid organs in a couple of important ways. One, it functions strictly in T lymphocyte maturation. Okay, the T lymphocytes mature in the thymus gland. That's why they're T lymphocytes. T lymphocytes meaning thymus. Okay, it does not directly fight antigens. The lymphocytes will, but the thymus glands do not directly fight antigens. Okay, the stroma of the thymus consists of star-shaped epithelial cells, but there's no reticular fibers. Okay, and these thymocytes will secrete the hormones that stimulate lymphocytes to become immunocompetent and be able to help with immunity of the system. Let's check out the thymus. Okay, on the left we see the individual here, and we see that it's exposed and enlarged, and we see that the thymus gland is located on top of the heart. Okay, as you can see, it is found on the left side of the heart, right above the left atrium. Okay, one of the lobes there, whereas the right lobe of the thymus, okay, the bilobed organ, okay, kind of edges toward the right atrium, but most of the thymus, the left thymus, the larger portion, is on the left side. Okay. Okay, got a little overview of the lymphatic system here. What's going on? Okay, lymphatic capillaries are going to remove fluid from the tissues, and that fluid then becomes lymph. The lymph then will flow through lymphatic vessels, which then have valves that prevent the backflow of this lymph. Okay, then the lymph nodes then filter that lymph, and then where the lymphocytes respond to the infections. Then the lymph enters those thoracic ducts or the lymphatic ducts, and then the lymph enters the blood. And once that lymph has entered the blood, the lacteals in the small intestine will absorb that fat, which enters the thoracic duct. Okay, then the chelae which is lymphatic containing fats, then enters the blood. Okay, and since it contains that lymphatic tissue, okay, it's going to be able to be a little bit supported. Okay, the spleen then filters that blood and is the site where lymphocytes respond to infections and etc. So the spleen is a site in which actual attack of pathogens will occur by lymphatic cells. Okay, the lymphocytes, either pre-B cells or pre-T cells, originate from stem cells in the red bone marrow. Okay, the pre-B cells will mature in the red bone marrow itself. Okay, B for bone. Okay, B cells mature in the bone. Okay, and then they're released in the blood. Pre-T cells, those T cells will mature in the thymus. T for thymus. Okay, T cells mature in the thymus. And then the thymus is where the pre-T cells derived from the red bone marrow will increase in number and become mature T cells. Okay, and these are then released into the blood to help fight infection. Okay, then the B and T cells from the blood enter and populate all lymphatic tissues. The lymphatic tissues, or the lymphocytes, can remain in tissues or pass through them and then return to the blood. Okay, the B and the T cells can also respond to infections by dividing and increasing in number, and some of the newly formed cells can enter the blood and circulate to other tissues. What is immunity? Now we're getting to immunity. Okay, immunity is just not simply being able to get away with not doing some challenge on a game show. Immunity is very important. Immunity is the ability to resist harmful effects of microorganisms and other foreign substances on your body. Okay, adaptive immunity exhibits specificity. Okay, it is very specific. It does not span a broad region. It will attack individual antigens that present themselves. Okay, and it also provides a memory. However, innate immunity does not show specificity or memory. Adaptive is very specific. Innate, not specific at all. Okay, it does not show specificity or memory. Adaptive will show memory, and it is specific. Okay. Let's check out the immune response.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B.
Okay, remember, here's question number 3. Where are the cytotoxic T cells formed to maturity, and where do the B cells go to maturity?
Cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell.
Oops. Okay. Innate immunity. Okay, it responds quickly, and it consists of mechanical mechanisms where skin and mucosa prevent entry of microorganisms, and it's also found in the tears, saliva, and the mucus that removes them from areas. Okay. There's also chemical mediators, and there's cells, and then there's the inflammatory response that are part of innate immunity. Okay. Chemical mediators promote phagocytosis and inflammation. Okay, so you can see some of the things that work there. There's the complement system. Okay, the complement system is very intricate. The complement system is a group of plasma proteins that increase vascular permeability, stimulate the release of histamine, activate kinins, lyse cells, which means kill cells, and it promotes phagocytosis and attracts neutrophils, monocytes, macrophages, and eosinophils, which means all of the white blood cells. Okay, then you have cytokines. Cytokines are proteins or peptides that function as mediators of immune and inflammatory responses, and they're secreted by macrophages and NK or natural killer cells in innate immunity and by B cells and T cells in adaptive immunity. Then you have the defensins. The defensins are peptides, which means they're proteins that are produced by neutrophils and epithelial cells that disrupt microbial membranes. Basically what it means is it will break the membrane of bacteria cells and stop their ability to function or live within your cells. Okay, moving down. We got histamine. Histamine is an amine released from mast cells, basophils, and platelets. The histamine causes vasodilation, or swelling, okay, and increases vascular permeability, get a little redness, and it stimulates gland secretions, especially mucus and tear productions, and it causes smooth muscle contraction of airway passages in the bronchioles, in the lungs, and attracts eosinophils, specific white blood cells, for its actions. Okay, and then we also have the interferons. Interferons are proteins that are produced by most cells that interfere with virus production and infections. Then we also have the kinins, and the kinins are polypeptides derived from plasma proteins, and the kinins cause vasodilation, increase vascular permeability, stimulate pain receptors, and attract neutrophils. We also have the leukotrienes. The leukotrienes are the group of lipids that are produced primarily by mast cells, basophils, and that cause prolonged smooth muscle contractions. And these smooth muscle contractions increase vascular permeability and attract neutrophils and eosinophils. Okay, then we also have the prostaglandins. Prostaglandins are very important and are a group of lipids. That's right, they're a group of fats, the PGEs, PGFs, thromboxanes, and prostacyclins, which are some of those that cause smooth muscle relaxation and vasodilation. It also increases the vascular permeability and stimulates pain receptors, causes pain from prostaglandins. We also have pyrogenins. Okay, and the pyrogenins are chemicals that are released by neutrophils, monocytes, and other cells that stimulate fever production. So when you have the pyrogen, pyro means hot, so it causes your body to warm up in response to other invaders, and since those invaders don't function above your normal body temperature, usually the fever will in fact kill off those invading microorganisms. Surface chemicals. Then we have the lysozymes in tears, saliva, and nasal secretions that help break down bacteria and other invaders. That's also found in sweat as well, but mostly you find them in the other mentioned fluids. Okay, we also have the defensins, which are antimicrobial peptides produced by all epithelial cells. And we also have acid secretions. Okay, sebum in the skin and hydrochloric acid in the stomach. That's right, hydrochloric acid, a very strong acid, is found in the stomach of humans that prevent microbial growth or kill these pathogens. And mucus on the mucous membrane trap the pathogens until they can be destroyed. All right. So now we're going to hit each thing individually. Not all is one thing that's going to confuse you, just like that last slide did, but I knew it was going to happen, so we're going to individual parts. Now, complement system, very, very complicated as well. Okay, we're going to go with a complement system. And that complement system is a pathway that involves a cascade, okay, where things will increase in intensity as it moves along, in which complement proteins are activated in an orderly sequence. The end result is cell lysis, phagocytosis, and inflammation. Okay. The complement system refers to a group of 20 or so proteins that circulate in the blood in an inactive form, but however, there is a mechanism that complement will provide, a major mechanism to destroy foreign substances in the body. The complement system is very, very intricate, and we'll see that here in a minute. Okay, it amplifies all aspects of the inflammatory response when it activates the complement system. Inflammatory response caused typically by the complement system. Okay, complement system will also be able to kill bacteria and certain other types of cells. Okay, but our cells are immune to the complement proteins. Okay, more about the complement. And it can be activated by either the classical or the alternative pathway. The classical pathway of the complement system is part of the adaptive immunity, and it will depend on the binding of antibodies to invading organisms or the subsequent binding of C1 to the antigen-antibody complexes through complement fixation. Then we also have the alternate pathway. Okay, the alternative pathway is part of the innate immunity, and it's triggered by interaction among factors B, D, and P, and polysaccharide molecules present on organisms. So here we look at the complement pathway. The classical pathways we're going to look at first. Classical pathway is going to begin when an antigen-antibody complex binds to complement 1. Okay, the antigen-antibody complex then activates complement 4 or C4, and then activated complement 4 that complexes with complement 2 will activate complement 3. Yes, when we're talking about the complement system, 4 comes before 2 and 3. However, 2 and 3 go in order. Go figure, right? Who named them? Who numbered them? I don't remember, or do I even know? Okay, foreign substance and factors B, D, and P will then stabilize the activated complement 3. Once complement 3 is activated, the classical and alternative pathways are then the same. So the area in which the pathway from C3 on is initiated is the same as the alternative pathway. So they start off differently, but the complement pathways end up with the same activation levels once they've reached complement 3 activation. Okay. Complement 3 through 7 will promote phagocytosis, or cells being eaten or engulfed by white blood cells or lymphatic cells. Okay, phagocytosis, inflammation, as well as chemotaxis. Okay, chemotaxis is the emitting of chemicals in the body that will attract other cells to come to that area. So when it emits the chemotaxis response, it's going to recruit other lymphatic cells, other white blood cells to come in and help fix the problem. Okay, activated C5 through C9 or complement 5 through complement 9 will combine to form a membrane attack complex, or the MAC complex. MAC complex is the membrane attack complex, and this membrane attack complex forms a channel through the plasma membrane. Only complement 9 of the MAC is shown here. See it right in this area. Okay, interferons. Interferons, we're moving into another little area here. Okay, complement system, we've done all we're going to do for right now. We're going to interferons. Okay, interferons, what will happen here is they will leave the infected cell and enter neighboring cells. Okay, interferons will stimulate the neighboring cells to produce the proteins to prevent the replication of viruses. Okay, so interferons help to prevent virus formation or viruses from reproducing. Okay, interferons will activate macrophages and the NK or the natural killer cells. Okay, here's some cytokines and their functions. So we got type 1 interferons, interleukins, and then we got TNF, the tumor necrosis factor. All right, I want you to be very aware of each different type of cytokine. That's why we're only doing one chapter this time. You need to know the immune system very well if you're going into the medical profession. All right, so here we look at the type 1 interferons. Okay, what happens here with the type 1 interferons is they will prevent viral replication, increase natural killer cells. Okay, you can read the rest of it yourself, but I want to go over the tumor necrosis factor for sure. Okay. Tumor necrosis factor, what happens here is it recruits neutrophils and monocytes to the sites of infection that activates them. Okay, it also promotes fever or pyrogen to come in, and that pyrogen response creates an increase in temperature, will kill off some of your invaded cells, and in large quantities contribute to septic shock, and it can be secreted by macrophages. Tumor necrosis factor, okay? Talking about it here in the immune system, but tumor necrosis factor also will help your body deal with your own cells when they come out of control and start to form cancers. As a matter of fact, most of us have these things happen often, and our tumor necrosis factors, which tumor necrosis, which means tumor killing factor, okay, necrosis or death. Okay. These tumor necrosis factors typically will wipe out the formation of these cancers, but every once in a while your system gets overwhelmed and you actually end up with a cancer which will affect your body, not one that you have cancer for a little while and your body takes care of it and you never even really know you had a problem. Okay. Cells in the innate immunity. Okay, chemotaxis is the ability of the white blood cells to move to tissue and then release certain chemicals. And then we also have phagocytosis, which is the ingestion and destruction of materials within the white blood cell itself. Neutrophils are small phagocytic cells. They're very small phagocytic cells. Okay, and then you have basophils and mast cells that release chemicals that promote inflammation. Okay, eosinophils that release enzymes that reduce inflammation, and then your natural killer cells that release enzymes that lyse tumor cells and virus-infected cells. So they actually kill your cells containing tumors or virus-infected cells, natural killer cells. Okay, macrophages. Remember, neutrophils are small phagocytic cells. Macro, or large phagocytic cells. Okay, and these large phagocytic cells can engulf more than neutrophils can by far. Okay, and in connective tissue, they protect the body at locations where the microbes are likely to enter. And macrophages can then clean the blood and the lymph tissue. Some macrophages can even have specific names. Okay, dust cells are found in the lungs that help destroy some of the dust that comes in, which contain particles of bacteria. And then you also have the Kupffer cells in the liver, as well as the microglia in the central nervous system. So here we see the immune cells and some of their functions, and we look at these, and we got the neutrophils that we talked about, monocytes, macrophages, dendritic cells, natural killer cells, the mast cells, the basophils, the eosinophils, and those found in the adaptive immunity. Remember those mentioned before are part of the innate immunity. Neutrophils, monocytes, macrophages, dendritic cells, natural killer cells, mast cells, basophils, and your eosinophils. And then you go to the adaptive immunity, those that are specific. Remember, innate is not specific. Adaptive is specific. Okay, and it also has memory over time. You got your B cells, your plasma cells, memory B cells, cytotoxic T cells, your helper Ts, and your memory Ts. Your memory Ts are ones that are going to remember for the longer period of time or exactly have a specific attack spot over a longer period of time. Okay, inflammatory response. The inflammatory response of the body can be initiated in many different ways. Okay, chemical mediators can cause vasodilation and increase vascular permeability, which allows the entry of chemical mediators. Chemical mediators then attract phagocytes, cells that are capable of phagocytosis. What were they called? Remember, you got the macrophages, and you also have the neutrophils. Okay, the amount of chemical mediators and phagocytes increase until the cause of the inflammation is destroyed. Then the tissue then must be repaired. Okay, the inflammatory response. Local inflammation produces symptoms of redness, heat, swelling, pain, and loss of function. Okay, and symptoms of systemic inflammation will also include an increase in neutrophil numbers, increase in fever, or in extreme cases, you would even end up with shock. Okay, so figure 19.10 or 19.10 breaks down what would happen if you have a bacteria invader. Okay, tissue damage is occurring due to this bacterial invader. Okay, chemical mediators would then be released by the body through the system. Okay, chemotaxis causes increased vascular permeability. Why vascular permeability? That means that cells can move in and out of the blood vessels so that they can attack or move to the areas in which they are needed. Okay, and this will also help to increase blood flow, because if the vasodilation occurs, the red blood cells can even move better. The complement can move better. The white blood cells can move through better. Okay, and the increased number of white blood cells in the chemical mediators at the site of tissue damage will increase. Okay, the bacteria are then contained, destroyed, and phagocytized. Okay, and if this works and the bacteria are gone, the tissue is then repaired. If this doesn't work and it's not all done, the bacteria still remain, then additional chemical mediators are activated, and this loop that you see on the right side happens, and the process occurs over and over and over again until the situation is stable. Adaptive immunity. Okay, adaptive immunity is a functional system that recognizes specific foreign substances that enter the body, and it acts to immobilize, neutralize, or destroy these foreign substances. It'll also amplify inflammatory response and activates the complement system. The adaptive immune system is antigen specific, one. It is systemic, and it also has memory. Okay, three things there. Okay, the adaptive immune system is antigen specific. It is systemic, and it has memory. That makes it very different from the immune system? Okay. It has two separate but overlapping arms. The antibody immunity, okay, antibody-mediated immunity, which is provided by antibodies in the blood and the lymph flow, or the lymph tissue, okay, and the cell-mediated immunity, which lymphocytes become involved themselves, okay. Adaptive immunity, we're going to go in a little bit further. We're going to talk about antigens, how are antigens formed. We're going to talk about that. Okay, antigens are large molecules that stimulate an adaptive immune system response. Okay, an adaptive immune system response, which means it's going to send in something to remember, one, and attack, two, and remove the threat, three. Okay, foreign antigens are not produced by the body. These are not self-antigens. Okay, self-antigens are produced by the body. We're talking about foreign antigens like bacteria cells that come in. B cells are responsible for antibody-mediated immunity. T cells are involved with cell-mediated immunity. What's the difference? Okay. Origin and development of lymphocytes. We've talked about this a little bit before. B cells and T cells both originate in the red bone marrow. They're both produced there. But the B cells are processed or they mature in the bone marrow. Whereas the T cells are processed or mature in the thymus gland. So the T cells basically absorb the name of the thymus and they're called T cells. The bone marrow provides the name for the B cells. Okay. The positive selection ensures the survival of lymphocytes that react against self-antigen, or that can read against antigens, and then you also have the negative selection, eliminating lymphocytes that will react against self-antigens. Okay, a clone is a specific group of identical lymphocytes that can respond to a specific antigen. B cells and T cells move to lymphatic tissue from their processing sites, and from these processing sites, they continue to circulate from one lymphatic tissue to another. So here we can look at the circulation, how it's happening. See, they both come from the red bone marrow. We're looking at the bone up at the top. See how they move to different areas like the lymph nodes or the thymus gland. Okay, in the thymus gland, you got your T cells maturing. Okay, you got in the bone marrow, they're maturing in the bone marrow itself, so they just move directly to the lymph vessels or the lymph nodes. And from those lymph nodes, they can move to different areas which are needed through the circulation of lymph tissue and adaptive immunity. Okay, the activation of lymphocytes have an antigenic determinant or an epitope. An epitope is the specific part of the antigen to which the lymphocyte responds. Basically, it's the part that's remembered or the part that's recognized as being an invader on the antigen. Okay, the antigen receptor, the T cell receptor or B cell receptor on the surface of lymphocytes combines with the antigenic determinant. Okay, the MHC class 1 molecules will display antigens on the surface of nucleated cells, resulting in the destruction of the cells. However, the MHCs, for the major histocompatibility complex, which is what MHC stands for, major histocompatibility complex, class 2 molecules display antigens on the surface of the antigen-presenting cells. And this results in the activation of immune cells. Let's check out antigenic determinants. Look and see what an epitope is. It's probably going to be easier for you to see than me to put it into words.
Antigens are macromolecules, usually of molecular weight greater than 10,000, such as proteins and polysaccharides. They are recognized by the immune system as foreign. Individual antibodies are not made against the entire antigen molecule, but rather to particular chemical groups on the molecules known as antigenic determinants or epitopes. Many different antibodies can be made against a single antigen, each antibody reacting with a different epitope. Complex structures, such as the surfaces of bacterial cells, may have many different epitopes. Each different antibody binds only to the correct epitope.
Okay, so we saw that each antibody will only bind to a certain epitope. So basically, antibody recognizes the epitope and directs the complement system, or directs the activation of the other systems. There we go. Alright, so we see how this moves in different areas in major histocompatibility complex. We've got foreign proteins or self-proteins within the cytosol or the cytoplasm, broken down into fragments that are antigens, and these antigens are transported into the rough endoplasmic reticulum, and from there we see that the antigens combine with the major histocompatibility complex 1 molecule and an MHC class 1 antigen complex is transported to the Golgi package and then kicked out, basically. Okay, the foreign antigens combine with that major histocompatibility complex, and then cell destruction occurs, and then that self-destructing activity, okay, excuse me, does not stimulate the cell to destruct, but it stimulates the release of those contents to the exterior of the cell. Okay, down at the bottom we got a different type of situation going on. We have an MHC class 2 molecule coming in, and the major histocompatibility class 2 molecules are going to act a little differently. So we have a foreign antigen that come in, and it's ingested during endocytosis, and is within a vesicle within the cell because it's been encapsulated by the membrane. Okay, and that antigen is broken down into fragments to form processed foreign antigens, and that vesicle containing these foreign antigens fuses with the vesicles produced by the Golgi apparatus that contain the MHC class 2 molecules. And these antigens then combine with the MHC class 2 molecules and then they're transported to the plasma membrane. And then it can stimulate immune cells to come and say, hey, take care of these things here. Well, you can't do anything with them. Alright, so they come in and destroy these invading antigens. But they provide that signal by moving to the exterior of the cell. Alright, let's check out.
Antigens are processed differently, depending on whether they originate within or outside the cell. Proteins produced within the cell, such as viruses or self-proteins, are broken down into fragments. Fragments of foreign proteins are antigens. The antigens are then transported into the rough endoplasmic reticulum. The antigens combine with class 1 MHC molecules. The combination is then transported to the Golgi apparatus, and from there to the plasma membrane. Foreign antigens presented on class 1 MHC molecules stimulate cell destruction by activated T cells. Self-antigens are not recognized by the T cells and do not stimulate cell destruction. A different process occurs if the antigen originates outside the cell. Phagocytes, such as macrophages, ingest foreign particles such as viruses and bacteria by endocytosis. The foreign particles are broken down into fragments within a vesicle. The vesicle containing the foreign fragments fuses with vesicles from the Golgi apparatus containing the class 2 MHCs, and the two structures combine. The MHC class 2 antigen complex is transported to the plasma membrane. The displayed MHC class 2 antigen complex can stimulate other immune system cells to respond to the antigen.
Okay. All right, everybody. This is going to be the end of part 1 of the lymphatic system. I'm going to post a part 2 shortly. You guys have a good evening. And study hard, and hopefully your grades stay well. And for those that need a little improvement, hopefully you will improve shortly. Hopefully you guys were pleased and got what you expected at your midterms. There were a couple of you that need a little improvement. Others are doing great, and I'm very proud of you guys. Hang in there, and I will update as soon as possible. Thank you guys.